So histone methylation, I'm here. Histone. So the loosening and the tightening of chromatin. Here I'm going to discuss about the two different enzymes, a type of enzymes. Exactly, this is four type of enzymes. Sorry for that. So the first one is your histone acetylase. So what it do? I mean the here you see the repression and activation of the control of a large system. Forget about all those things. Let me just clarify you. There are two things. One is your methyl group, this is your CS3 group, and one is your acetyl group, COCS3 group. So both these groups play a very important role in controlling the expression of a gene by repressing or activating its function. So let's first discuss about your acetyl group. Acetyl group is your COCS3 group, which is the -I group. So what happened? So when it goes and binds with that histone molecule, so the histone is, as we know, it's your positively charged protein because it possesses lysine, arginine like basic amino acids, whereas your DNA is your negatively charged nucleic acids, so it better bind with them. So when your acetyl group goes and bind with that of your histone, and as your histone this acetyl group is negatively charged, so it neutralizes that histone positively charged, and that neutralization cause DNA molecule which are coiled around this histone becomes free to get activated and undergoes transcription. That's the important thing. So which enzyme do this kind of stuff? Well, this is your histone acetylase, or what you call, sorry for that, histone acetyl transferase. This histone acetyl transferase which transfer this acetyl group, this negatively charged COCS3 -I group, to get attached with the lysine residues of that histone H3 protein, and there it neutralizes its basic charge and release this DNA from its coiling and being able to transcribe. And another enzyme is your histone deacetylase, which do the opposite thing. That means it removes this acetyl group from that region, and once again it come back to a repressed state, so genes become unable to transcribe. So next, your histone methylation. Methyl is your +I group compound. That means it provides the electron. That means it is your positively charged group. So when it goes and bind with that your histone molecule, there it does that is, as it is a positively charged and histone is also positively charged, being basic, so they make this DNA, which is negatively charged, more coiled tightly, and that causes repression of the DNA. That means the DNA is unable to transcribe. That's the thing. So this is how the two things act antagonistically. The acetylation causes the activation of your DNA, whereas your methylation causes repression of the DNA transcription. So this is up to the methylation of the histone. I'm not talking about the DNA methylation, I'm talking about the histone methylation and acetylation, histone protein, because later on we're going to discuss about the same thing but on the DNA molecule. So the other things, and one more thing is that whenever the methylation is done in your this fourth lysine residues of your H3 tail, histone 3 protein tail region, it generally don't what we call repress this DNA. Rather, it play a other type of role, not that kind of, but it's like neutral role or little bit positive role in activation of transcription. That means active expression of your gene. But the same methyl group, if binds with different lysine amino acid of 9th region or 27 region or 20 region of that histone molecule, then it causes this methylation causes repression of that chromatin. So this is the thing. But and the acetyl group when bind with that 3 or 4 region of the H3 or H4 protein, it causes activation. So this is the thing. So here you can see the picture that whenever the H3 and H4 tail is methylated, it get repressed. That means the condensed nucleosome, that means the nucleosome get condensed, tightly packed, so it unable to go transcription. But whenever it get unmethylated and get acylated, that means COCH group get attached with that histone tail of lysine residues, then it cause it to free rather being tightly packed, so it's easy to transcribe those genes. So this is the difference. Here the tail which methylated is your H3 and H4, and here in this region your S2 H3 tail are being normally this your acyl related, and H1 is your linker, so it's not present here. I'm not shown here, sorry, not present, it's present always, but not shown here. So this is the thing, and you can see here different kind of activation is regulated by different region of that methylation of tail region. Now one more thing is also important is the maintaining of memory of methylation. So there are two protein like your trithorax and polycom, and I'm not going very deep in deep. Two things, remember, the trithorax and polycom is like a memory that they can memorize that methylation pattern and keep it in methylation. So trithorax the one which keeps the gene active. And how does it works? Well, it binds to the condensed nucleosome and keep the gene in a repressed state. The protein keeps gene active where the trithorax protein keeps the gene active, where the polychrome protein keeps the gene inactive. So the trithorax protein inhibits those binding of your methyl group and keeps those genes unmethylated histones, and this is how the trithorax keep the particular gene active. And this is really helpful during this oocyte condition where the multiple gene are need to be expressed, and that has some memory. So on further differentiation this is helpful. Okay, so trithorax is always the protein which keeps the gene active, while the polychrome is always the gene which repress those gene expression. Here written, you can read it. I'm going to read out it. Trithorax protein can modify the nucleosome, alter the position and chromatin, allowing the transcription factor to bind the DNA previously covered out them. So trithorax, what it does, well, it is the position of the chromatin. The position of chromatin it gets shifted and allow the transcription factor to get bind on it, and that's how it's kept active. Whereas your polycom which bind to methylated tails of histone 3 and keep methylation active and also methylated adjacent nucleosome, thereby forming the tightly packed repressive complex. This is how the polycom works. Now come to the anatomy of gene in next video, I think so. Thank you.